when the next candy surge arrives. That time it's cookies. No? Yes? Doesn't everybody like have a million cookies all over the place for Christmas? Do you need an extra chair? Here, let me share it with you. We're learning stuff. I'm sure there'll be stuff to learn. Is this yours? Okay. I don't know yet. I'm working on it. We're first going to ask a few more questions about this homework. Okay, hi. So, questions about homework? Yes, no, maybe. Nothing. All right. Put name on homework. Hand it to the left, hand it to the front. Okay. So, this week, what we're going to do is start working on some, guys, is this puny pile literally everything that people are turning in this week? Alright, that's slightly better. It is still a puny pile, y'all. Alright, so here's what I'm recommending. Chapter 16 and 17 are really crucial chapters, as is 18, as is 20. These are chapters that are going to get questioned a lot. Oh yeah, we have more. Thank you. Alright. What are we talking about right now? Oh, crucial chapters, 16, 17, 18, 20. Crucial in the sense that if you were to pick up a textbook, not even a textbook, if you were to pick up a paper about the kind of science people are doing nowadays in cell molecular biology, those chapters are at the basis of understanding what's going on. So we're going to go over some of 20 this week, some of 20 next week. But when it comes to doing the chapter 16, 17, 18, and 20 practice, don't skimp on that. It's worth your time. It's also very important for the Usabo. Alright, because Usabo will ask you weird, complicated questions about self-molecular stuff. What are we on right now? I don't know. I haven't started yet. Give me a minute. I'm warming up. Alright. So, let's do this. In pairs, work with your neighbor to explain transcription and translation, including all of the enzymes. And be able to contrast RNA polymerase with DNA polymerase. So transcription and translation, all the steps, all the enzymes, and be able to contrast RNA polymerase with DNA polymerase. Take 5 minutes, please.
Yeah, so already the
No, I'm pretty sure those all
DNA polymerase
What's the thing that prevents the poly?
The A site? Yeah. I mean, it's single amino acid.
Yeah, I feel like there's a sensory.
Yeah. Yeah.
Anything that made Anybody not playing any Alright, it's going around the second time. I think you're thinking about DNA replication. What happens is RNA is put out for the DNA polymerase change. That's kind of the it's not the other way around.
No, yeah, but like, RNA, it's like, oh sorry, RNA is
and then it gets taken out by something else patches that.
Lysosome is RNA polymerase on mRNA. So like they're together.
I just did it because I like it. And then I figured out all the
Yeah, the cutout bit is the intron and then put together.
It's really fun though. Yeah. It's like the hair center.
There's something
Hairpin
that
is something else.
holds
Hairpin was in chapter 18 of microRNA. Hairpin is used
more
RNA.
Like a public.
Just like
Proteins that cut RNA.
when you make a polymer, something or you make two, like you make two atoms come together, but the atoms pick apart. Wait, does anyone else need to sign in?
How many EPs could you make and how many EPs could you make?
Questions? No. Okay.
Does anyone else need to sign in? You need to sign in.
Who else needs to sign in? Alright, over here. Questions back here? Alright, questions in the second row. Now we're awesome. Questions in the first row. Alright. So we have reviewed stuff. So, did anybody try to start explaining transcription with a helicase? No? We're good? We know transcription starts at the promoter using transcription factors, which attract the RNA polymerase. Do we all remember what the Tata box does? Who can tell me what the Tata box does? Yeah.
Is it where the RNA polymerase binds to should start transcribing?
Exactly. The Tata box is a sequence of DNA that helps RNA polymerase bind to the right spot. That's right. How is RNA polymerase different from DNA polymerase? Yeah, well, no, I want them to answer it, sorry. RNA polymerase different from DNA polymerase? Yes.
You can just add nucleotides on top of the template strand instead of like having to have like a primer.
Exactly. The RNA polymerase doesn't need a double strand primer. It can just straight up start adding. Good. What's another way?
It can separate the two strands of DNA
Yeah.
without using like another enzyme to do it.
Exactly. RNA polymerase can separate the strands of DNA without needing the help of helicase or another enzyme. What else? Yeah? Polymerizes RNA instead of DNA. Important difference. Extra credit. How are the RNA nucleotides different from the DNA nucleotides?
So there's single strand and also
Instead of T, it's U. Single strand instead of double strand. And, yeah?
Instead of using deoxyribose as a sugar, it uses ribose.
It uses ribose instead of deoxyribose for RNA. Good. What is similar between RNA polymerase and DNA polymerase? Well, what is their job? Yeah? Yeah, they're both adding complementary nucleotides. They both need a template. They're adding complementary nucleotides. They're creating sugar phosphate bonds to create a sugar phosphate backbone. They both have a triphosphate coming in. They take off two of the phosphates to create the strand. And they both go from 5' to 3', meaning the new strand we're adding to the 3' end. All right, Melissa. Do you remember what a plasmid is? Yeah? Yeah, what's a plasmid? Yeah, so it's a small piece of circular DNA that's outside the chromosome of a bacteria. All right, now, we will be talking about chapter 20. Don't bother, if you want to open the book to chapter 20, go for it, but we're not going to talk about specific figures just yet. I'm going to give you kind of a more big picture understanding of how we do science. Yeah, modern science isn't just the scientific method. Sure. All these markers are terrible markers. Yeah. Old stuff to hand back for this class? For AP. Okay. Got it. So, okay, that works. So let's say you want an organism to have new genes, right? As a scientist, if you're trying to explore the function of a new gene, or a function of an old gene, a function of a gene, you want to be able to see what a cell does if it has more of that gene, what it does if it has less of that gene. That's the easiest way to check out the gene's job, right? And then you want to look at it in an organism, not just in a cell. What is the organism like with more of the gene? What is the organism like with less of the gene? So you already know one method of making less of that protein that the gene codes for. What's that method?
RNAi.
RNAi. In a cell, you can add the correct hairpin turn RNA to that cell, and in response, the cell will use that hairpin turn RNA as a template for destroying mRNA, and they'll be specifically destroying the mRNA that codes for your protein of interest. We'll be talking a lot about poise and GOIS. Protein of interest, gene of interest. Okay? I'm not saying that you will always see them written like this, but they will be set. Protein of interest, gene of interest. So now, here's your cell. Bob the cell. Here's your hairpin turn RNA, not to scale. How would you stick this in here? What can you do? Yeah.
If Bob's a prokaryote, you can add the hairpin into a plasmid.
Yeah. Ooh.
Or incorporate it with a plasmid.
Yeah, you could have a plasmid that's DNA that codes for RNA that would create that hairpin turn. Yeah. Okay, here's a plasmid. How'd you get the plasmid into Bob? With my finger? Yeah? Ooh, so there was a suggestion that we could use conjugation. Conjugation, who remembers what conjugation is? Yes?
The bacteria extends its pili so it can transfer the DNA into the other bacteria.
So Bob already has the plasmid and he sends a copy of it over to Bill, right? But, again, we'd have to put it into Bob, is the problem. So we have to put it into the one bacterium before I can move it around. Other thoughts? Yeah?
Shock it with electricity.
We can shock a bacterium with electricity. When bacteria are in stressed situations, like electricity or a big change in the temperature, their membranes become a little bit more porous and DNA can get in. They are more likely to be transformed. Transformation is how bacteria naturally pick up DNA from their environment. So if we have a prokaryote, we can transform it literally by shocking it with electricity. We call the process of shocking it with electricity electroporation because we make it more porous with electricity. And I mean, it sounds really cool. Like, it sounds like you're like Frankenstein in the lab going, "Mwahaha!" And lightning goes down to your little bacterium and he's like, "I'm alive." But it's not like that. You put your bacteria with your plasmid in like this tiny little, it's like a test tube, but it has metal on both sides to help conduct the electricity. And then you stick that into a machine and then you push a button. And then the machine sends electricity through it. Kind of like you flick a switch and it sends electricity to the lights. Not that interesting. Not like Igor, pull the lever. Like, that doesn't happen. No, man, if I worked in a lab with somebody called Igor, oh, the jokes. So that is how you transform a prokaryote. How else? Do you guys remember what's the third way we have horizontal gene transfer in prokaryotes? Yeah?
Transduction.
Transduction, which means we're using what?
Virus.
Virus. Good. Transduction, which uses a virus. So if you somehow put the gene you want in a virus, you can then use that virus to put the gene in cells. Virus are like pre-made cell-invading, gene-inserting machines, and they don't just do it to bacteria, they do it to eukaryotes. So here's my question for you. How can I make a virus that has the gene I want inside it? Where do viruses come from? Hosts. And how do the hosts know what to do to make the virus?
DNA.
DNA. The virus injects DNA, the DNA tells the host what to do, the host makes more virus. So if I took a regular, like, virus DNA, and I modified that virus DNA, maybe I keep the parts that are necessary for creating the viral envelope, but I take out the parts that I don't want, or maybe add some new parts. And the new thing I've added is the gene I want, my GOI, right? My gene of interest. Then I put this in a plasmid, and I electroporate that plasmid into Bob. And then what happens with Bob? Bob uses that plasmid to start making a bunch of viruses that have my GOI inside them. And those viruses explode out, and maybe they, infect the nearby Bob Juniors, and then they make a lot of GOIs. And so by the time I'm done with that whole process, I have a little plate or test tube that is full of viruses that have my gene of interest. And then I could, if I wanted to, stick those viruses into a eukaryote. Depends on what the virus is, right? Some viruses don't go into eukaryotes, some viruses do. If it's a virus that goes into eukaryotes, they'll be able to get it into a eukaryote.
So in treatments, people don't necessarily use lytic viruses, right? Because then it would just like kill all the cells in the body, because it would just keep producing the virus.
Well, we're not actually trying to treat a human with a virus yet.
Oh,
We are primarily trying to use that virus to get genes into cells that are in a petri dish.
oh,
Yeah. That said, here's a word we need to know, vector. So if your vector is a plasmid, it's a plasmid vector. You put it into the cell, it's a vector that brings the gene in. But you can also have a viral vector that brings the gene in. Sorry, yeah.
I feel like it's kind of like using that like a transportation machine.
Yeah, a transportation machine. Your transportation machine could be a car or a bus. It could be a plasmid or a virus. Now, back to answering your question. Yes, we might want to break the virus's ability to finish the lytic cycle,
okay.
right? That viral DNA, we probably took out the part
Oh,
that makes a lot of it and it bursts out of the cell. We probably just kept the part where, it can do it once. Or maybe, the rest of this is like, okay, if I get this in, it'll do it once and burst once, but then the actual bit that goes into the new virus is just what I want without the lytic part. Like, we can do all sorts of stuff to it. Once we understand it.
Right.
Yeah. If it was you, how would you understand the jobs of the different chunks of DNA on a viral vector? What could you do? Yeah?
You could use DNA express one like a particular segment of DNA more than another, so then you can see like how it affects the changes.
Right. If I have my original viral sequence, I can cut out this part and try to put it into Bob. Or cut out that part and put it into Bob. What happens if I selectively delete different parts of it? And if I'm intelligent, if I'm like, okay, I'm just going to delete like 50 base pair chunks, and then we're going to delete 100 base pair chunks. Base pair is pairs of nucleotides. I can really learn what each chunk is responsible for, which is very helpful. Right, so one way I can get my eukaryotic cells, what should we call our eukaryotic cells?
Harry.
Eunice, is that a good name? So we got Bob the bacteria and Eunice the eukaryote. Eugene is nicer, okay. Eugene. I don't know how to spell Eugene, that's how I'm spelling Eugene right now.
That is
That
spell
is how you
Eugene.
spell Eugene. So he has good genes? Really? Like that's what the name means? Okay. Anyway, so as I'm to put DNA into Eugene the eukaryote, I can use viruses. But you just learned it's quite the process to make a viral vector that has the DNA you want inside it. It's annoying. It's actually a much easier process, and we'll talk about how to do it today, of making a plasmid and try to get that plasmid into Eugene. But as you know, Eugene wouldn't normally undergo transformation. It's not a thing Eugene does. So what we're going to do is we're going to do something called transfection, like transformative infection. What we're going to do is we're going to add a special lipid to the plasmid. So you have your plasmid, P, and you're adding a special lipid that surrounds, I'm using the letter L to indicate lipid. You add a special lipid that surrounds your plasmid. And because it's surrounded by a lipid, now that little lipid droplet can fuse into membranes and allow the plasmid into that cell's cytoplasm. And so, you can just use a plasmid and a special transfection agent, like a variety of different lipids, to get it into cells. Woohoo! Now, real question, what if you wanted to get the gene into an organism. Does a virus ever infect all of your cells?
No.
No. Even like really, really good viruses, like you develop an immunity, you know, they don't get all your cells. So a virus isn't going to work very well. The best way to make sure a gene is in all the cells of an organism is to put that gene into the zygote, the first cell. And you don't do that to humans because most of those zygotes die. Well, yeah, obviously illegal and unethical. And the reason it's illegal and unethical, one of the reasons is you don't want to go killing zygotes all over the place. Not unless other animals' zygotes are their problem, okay?
We don't care, that's the exact thing.
Yeah, I'm joking, but yeah, clearly we don't think very highly of other organisms as zygotes. I mean, we eat them all the time, right?
Yeah.
That's true.
Eggs,
Chicken.
chicken. Well, it's not a zygote anymore, but it's an organism. We eat
Oh.
it. It's delicious. Fish roe. Fish roe. You know, little fish eggs, like that little red stuff on your sushi. That's fish zygotes.
So what about caviar? Like,
The
Caviar, yeah. Lots of fish zygotes. We eat them all the time. Anyway, guys, off topic. Let's get back to this. Thank you. Where I was going with this is, most labs have like a low effectiveness. Like if they go with 100 mouse zygotes, maybe 5 to 10 are going to take the DNA in. And they have no way of predicting where in the genome the DNA is going to insert itself. Now, I want to remind you of something you already learned in chapter 15. In chapter 15, there was a section about chromosomal mutations, and they were talking about how, you know, you can have an inversion where this chunk of DNA just flips around on itself. Yeah? And we said that's actually really bad because the location of a gene on the chromosome determines how much of it you make. The location on a gene of a gene on a chromosome relative to other nearby promoters is going to control not just how much you make, but in which cells you make it. And so, for example, I was recently looking at a study where they were trying to study neurons and give them like rainbow colors. It was called Rainbow. I recommend Googling Rainbow. It's a great article. Anyway, they had like 19 mouse lines. And then if you look at every mouse line, some of them, like 5 or 6 of them, had great like neurons having colors, but then some of them had other types of cells that also had the colors. And the reason for this variation between the mouse lines is where in their genome did the new gene insert. It changes the expression. Location changes expression. This is important. Location and promoter change expression. So if I was to do this on a human, if I were to be like, hey human, you seem to have sickle cell anemia. I want to change your genome so you have the gene for normal hemoglobin. Well, what am I going to do? If I just stick it into you as a zygote, I have no control where that hemoglobin is going to insert itself into your DNA. It could insert itself right in the middle of a tumor suppressor gene, thereby inactivating the tumor suppressor gene and, you know, giving you cancer. Right? Chapter 18, reference. It could be inserted right next to an oncogene and somehow make that oncogene expressed more. Also bad. Or it could, based on its location, itself be expressed too much or expressed too little. So yeah, again, this is why we don't do it with humans. You don't do medicine with that big unpredictability of results on a human. You guys have heard of gene therapy, right? The idea is we want to fix your gene. If we know that a gene is making you sick, but we have had to be incredibly careful with it because early attempts, I'm talking in the early 2000s, late 90s, one person got cancer, exactly, out of 5. 1 out of 5 getting cancer, that's not the rate you want. You want 0, right? Okay, so we got this. Let's start zooming in to the actual tools we have as scientists to do these experiments. Now, I recognize that when I was your age and I was reading chapter 20, I just fell asleep and gave up. That is why, as your teacher, I'm going to separate chapter 20 over multiple weeks. And I'm going to do my best up here to help you really visualize what you're learning and see how it's relevant to a scientist. But yeah, I get it. I didn't really get what was in chapter 20 till I did it personally myself. You know, graduate school, so. So if you're like, it's so hard, that's okay, it's so hard. It's like that bit in Shrek with the triangles. Okay. Except this I actually use. Seriously, who thinks they're going to use that triangle stuff? Like ever. Unless they're an architect or something.
Not competitions, yeah. Only.
Alright, no, fair enough. Architects need it, engineers need it, anybody who's creating anything.
Oh wait,
needs it.
physics, right?
Physicists use it. Yeah, not physicians. Physicians do not use it. Physicians use what you're about to learn right now. So we're going to start with a technique called PCR. This technique called PCR, that's short for polymerase chain reaction. Okay, who knows and remembers all the steps of PCR? Less than a third of the class. So good, the rest of you get to learn, and a third of you get to feel smug. Don't worry, I'll knock you down later. I'll teach you something that you don't know.
Yeah.
The people already know it get to feel smug. It's their advantage for having learned it already. So, polymerase, like RNA or DNA polymerase. Chain reaction, meaning you use it over and over and over and over and over and over and over again. What does that mean? You do a lot of replication. PCR is a way of making lots of copies of the DNA you want in a test tube. And we're talking like really little test tubes too. So you're going to need some water because, you know, reactions happen in water. You're going to need the DNA that you want to replicate. So we're going to call it your template DNA, right? Where do you get your template DNA? I don't know. What are you experimenting on? Are you trying to study human DNA? You probably got it from a cheek swab. Or maybe from like blood droplets at a crime scene.
Damn.
Actually, blood does have DNA in it, just not a lot. It has white blood cells. But hey, common trick question. Red blood cells don't have a nucleus, therefore they don't have DNA. If you're trying to study red blood cells for genes, you're in trouble. It's not going to work. However, white blood cells do have a nucleus, and therefore do have DNA you can study. So yes, you can still get some DNA from blood at a scene, just not a lot. Okay, maybe you are studying the DNA of a virus, or a bacteria, or of a cricket. Doesn't matter. You took a little bit of that organism, you mushed it up, you isolated the DNA, or maybe sometimes you didn't even isolate the DNA, you just put the mush right in here. It has some DNA in it. And then, you don't want to replicate all the DNA. Because if you replicate all the DNA, it's going to take forever, there's going to be more likely to have mistakes, and there's going to be a lot of DNA in there. PCR is about replicating specific DNA. So, as you know, DNA polymerase has to start wherever a primer is. It can only start where there's a primer. If there's just a single strand of DNA, it can't. So you're going to have to add primers that are for the beginning and the end, of the strand of DNA you're studying. Does that make sense? How can you make sure your primer is for the DNA you're studying? Let's think about it. Let's say your primer is 2 nucleotides long. So maybe your primer is A. And let's say you know your DNA is 1,000,000 nucleotides long. How often do you think AA is going to happen in 1,000,000 nucleotides? A lot. Can we do the math on this? Well, let's think. For any one nucleotide to be A, it's a 1/4 probability. For the nearby nucleotide to be an A, it's a 1/4 probability. So 1/16 of 1,000,000 nucleotides should be AA. And 1/16 of 1,000,000 nucleotides is still a very big number, right? Okay, so what if instead of nucleotides, we went with 20 nucleotides? So that would be 1/4 to the power of 20, which I don't know off the top of my head, maybe some of you do, because you're clever in this class, divided by 1,000,000. A lot less likely to come up. A lot more likely to actually just be the one you want and not any random ones. Make sense? So here we are in the test tube. I have water, I have DNA template, I have primers, and obviously I'm going to need some sort of DNA polymerase, right? Oh, and because I want to make a lot of DNA, I need building blocks. What's the building blocks? Nucleotides. Dideoxy triphosphate nucleotides. So dideoxy nucleotide triphosphate, DNTPs. Lowercase d, the d is like an under like that. How do you say that? Underscore? Subscript. That's right. The D is subscript. Okay. So I'm going to need a bunch of DNTPs. Each of them needs to be a triphosphate. Dideoxy nucleotide triphosphates. What else do you normally need for DNA to replicate?
Primers.
Primers, we said. What else? Helicase, single-strand binding protein, topoisomerase, ligase, all sorts of stuff, right? Except you're not going to need any of it. This is all you're going to need because of the magic of science. Observe. You use helicase to break apart a double strand of DNA, right? Helicase breaks hydrogen bonds. But this is a test tube. All you got to do is boil it. If you boil it at 99 degrees Celsius for 1 minute, all those hydrogen bonds are just going to break on their own. Win! Skipped a bunch of enzymes. Good. By boiling. Then step 2, well, we need the primers to attach to the DNA. So we're not going to need primaries. We just let the primers attach to the DNA, but at 99 degrees Celsius they can't. We've got to make it cooler. If we make it too cold, all the DNA will start attaching back to the DNA. Right? So we don't make it too cold. We make it just cold enough. We make it like 72 degrees Celsius. Guys, that is hotter than you can touch. If you touch 72 degrees Celsius, it's going to make your hands red. And you'll be like, this is burning me. Right? This is Celsius, not Fahrenheit.
Oh.
Right? What's room temperature in Celsius?
Cool. 28.
Room temperature in Celsius? 25-ish, 28-ish degrees. Remember that. How warm are you?
38.
36. So 72 degrees Celsius, bad for your health. All right. So 72 degrees Celsius for like, I don't know, a minute, whatever. That is just cool enough for the short primers to attach, but not cool enough for really, really long pieces of DNA to attach. And then I need my DNA polymerase to work. Well, remember, you are 36 degrees Celsius. At what temperature does your DNA polymerase work? Yeah. So will yours work at 72 degrees Celsius? No. No. So you're going to need a special type of DNA polymerase from thermophilic aqueous bacteria. You're going to use Tech DNA from thermophilic aqueous bacteria that we found in Yellowstone Park in the hot vents. Their polymerase works at a hot temperature, 76 degrees Celsius, for 1 minute. Tech, T-A-Q, thermophilic aqueous DNA polymerase. TechPol. So we had the double strand, but we separated our strands. We annealed this was an annealing step. Annealing means gluing or sealing or attaching. And now we have a step for elongating the DNA using the Tech polymerase. And then we do the whole thing over again. We boil to separate our strands. We cool it off to anneal primers, and then we run it, we elongate it again. And then we do it again, and then we do it again. We do it again and again and again for about 34 times, give or take. Because every time, we're going to double the amount of the DNA that we have primers for. So we're going to double the amount of our GOI, gene of interest. In your textbook, this process is described. May I look in your textbook real quick? On page 404, figure 20.8. Page 404, figure 20.8. Guys, take a minute, look at that figure. Work with each other, make sure you understand polymerase chain reaction, PCR. You can use it, back here. Okay, questions? Yes.
So what was DNTP used for?
The oxyribonucleic triphosphate, or the oxyribonucleotide triphosphate. Questions? Yeah.
The solution, you have a time with your
Yeah.
and nothing happens to that because it doesn't have any.
Exactly.
Questions back here? Yeah, I'll give you guys one more minute and then we'll learn something else. Good. What's the note taking? The notes. It's mostly filling out a style. Are we okay learning more stuff? Are we ready? Do our brains have room? Alright, clear out some stuff. You don't need to know who the Kardashians did this week. Or what the Kardashians did. Or what their children dressed as for Halloween. Like, you're good. Just let the Kardashians go and focus on biology. People got my references here, right?
Yeah.
Okay, thank God.
Keeping up with the Kardashians.
If you didn't, it's okay.
It's a meme.
It's a good thing. Kardashians aren't a meme. They're a TV show.
I know, but like,
They've been memed a lot.
They're Kardashian memes now.
yeah.
They've been memed for a while.
There are a lot of memes.
Let's say you want to separate DNA by size. The reason I'm suggesting that you might want to separate DNA by size is that you just did PCR. And in your PCR, you had a bunch of your original DNA, which was very, very, very long chromosomal DNA. And then you have the new DNA that you just made, which is a much shorter chunk of DNA. Now, there's a lot of it, because you did PCR for a while, but you still want to separate this dude from all the other really long dudes, right? Strands, let's talk like grown-ups. Yeah? So how can you separate them by size? Yes?
Gel electrophoresis?
Gel electrophoresis. Who has heard of gel electrophoresis? Oh my God. Okay, I'm going to go over it quickly. But here is the mental metaphor I have for gel electrophoresis. Remember, imagine you have a piece of rope, and you're dropping that rope from the top of a tree. If the rope is really long and untied, it's going to kind of tangle on every branch and going to take a really long time to fall. If the rope is shorter, it's going to tangle less and fall more quickly. That makes sense, right? What I want to do with my DNA is I want to run it through a situation that's like a matrix of branches that are getting in its way and slowing it down. So the big piece is separated from the little pieces.
So it's like a sift.
What?
It's like a sift.
It's like a sift, but like multiple, like a sift with like, yeah, multiple sifts as it's falling down through that sift. Yeah. So what I'm going to use is something very much like Jell-O. Jell-O, for those of you who don't know, is gelatin. It's a protein. You get it from horses' hooves and bones. Yeah, you boil horse bones and you get gelatin. And then and basically what you do when you get gelatin as a powder from the supermarket, you add it to boiling water. And then as the water cools, it kind of holds a shape. The gelatin creates a matrix of cross connections. And those are kind of holding the shape. And then the water is kind of trapped in that matrix, and that's why your Jell-O is all jiggly, because it's water and protein, all connected. Now, as a scientist, you're going to make Jell-O, but not from gelatin. You're going to use a different protein called agarose. You get it from agar.
Oh yeah, from the seaweed?
From the seaweed, yeah. And you're going to use agarose, and when you add it to the boiling water, and then let it cool, it's going to solidify into a shape, and that shape is going to depend on the shape of your mold, right? If you make Jell-O in a cup, the Jell-O will have a cup shape. If you make agarose gel in kind of a mold that kind of looks like this, kind of like a rectangular shaped mold, kind of like the size and shape of your phone, then it will be a little gel that from the side looks like this, and the top looks like this, right? It's basically this long, thin, like rectangular gel. Make sense? Yes. Sorry. There we go.
Oh, agarose.
Agarose.
A-G-A-R. Agar.
I'm so sorry I spelled it wrong on the board. Thank you for correcting me. back to our gel. When we make that gel, we don't just put it into the mold. We put a comb in the mold. So if you put a comb in the mold, you're putting in something pokey, right, into the mold. When you take it out, you're going to have a bunch of little holes where the tines of the comb used to be. So when you're making your gel, you put in a comb and then you pull it out and then you have these holes. We also call those holes wells because we're trying to sound more fancy. The idea is if you're looking at it from the side, the well goes down, but not like all the way down. So there's some of the gel underneath it, and it's just a well in the gel. Okay? Now, you've got your liquid with DNA all inside it, and you're going to take that liquid and you're going to fill it inside the well. So all your DNA is in here. Right? So all your DNA is inside all of these wells. And then the gel itself, you're going to put it into a salty bath. It's inside a bath of salty water. And you're going to run electricity so that this is negatively charged and this is positively charged. Here's your negative charge, here's your positive charge. Through that salty water, because salty water conducts electricity well. And what's the charge of DNA?
Negative.
So which way will it travel?
Positive.
Towards the positive. And as it's getting pulled in the positive direction, it's going to get tangled in those branchings of the gel. Make sense? All right. Yes.
So then the shorter they are, the farther they travel.
The shorter they are, the further they travel. The longer ones will travel much less. Thank you, got it. And then, okay. And then, right, if they're the same length, they should travel on average the same speed. So everything in the same band must have traveled the same speed, must be the same length. If you got a little bit of a smear, then you must have a variety of length, and that's creating like a smear. Whereas if you have a really sharp band, they must all be the same length and traveling all at the same time. Cool. How do you see the DNA?
Colorful.
Well, you can't see it with a microscope. It doesn't zoom in enough. Yeah?
Ultraviolet light.
Ultraviolet light and, yeah?
You got a loading dye?
Loading dye. So the loading dye that you put in is actually blue, like, color, blue pigment, and it does not color the DNA. The loading dye travels on its own and it'll be like, ooh, here I am, because the dye is a molecule. It's a small molecule, it'll just travel through the gel like all the other molecules. The blue dye does not color the DNA. In fact, in the water and in the gel, you have a special molecule called ethidium bromide. Ethidium bromide. That ethidium bromide, by the way, can give you cancer. So don't drink it and don't splash it in your eyes. You'd be surprised what happens in labs. Alright, a student was telling me earlier this year that she didn't wear goggles and she got the gel squirting right in her eye. And she was like, so my eye was red? And I'm like, did you wash it? She's like, I washed it. Like, okay. You're still alive. You don't have eye cancer yet. This is good. But all I'm saying is wear goggles, don't drink it.
Safety equipment good.
Safety equipment good. Don't try to look all cool and skip the safety equipment. Just wear it. Just be the kid with an extra pair of gloves on. It's safer. Okay, so anyway, the ethidium bromide binds to the DNA, and in UV light, the ethidium bromide glows pink. So you put the UV light on your gel, and everything's like a little bit pink because it has ethidium bromide, but then the DNA itself is going to be a very bright pink. because the ethidium bromide bound to it and then the UV light makes it pink. So, boom, PCR, yes. Sorry, electrophoresis.
Like I did it before it showed purple. Is there like a purple light? Is there like something different going on there?
Well, UV lights have a little bit of purple coming out of them because they're ultraviolet, so they're going to have some violet and some ultraviolet, right? Yeah. Yeah.
Is there a alternative method that doesn't require UV light? Because I don't remember using UV light last year when I was in the lab.
I can see how there would be some other methods. UV light is the most common one. It's the one discussed in our textbook. Were you studying proteins or DNA?
DNA.
I'd be interested in looking at your methods sheet and seeing what you used.
I have it, sadly.
Like you didn't put it in a machine that kind of glowed?
I don't think so. It's like a plastic basin.
Yeah.
So it's in a plastic basin, and without moving it around or turning on any lights or anything, you just looked at it and could see the DNA? You sure you were seeing DNA?
I don't know.
What color was your DNA?
Wait, was the strawberry thing?
Could be.
Blended in, okay.
What color was the DNA?
Dark bluish?
Bluish. You sure you aren't just looking at your loading dye? Because the loading dye will give you bluish bands.
I don't know.
Usually it'll give you 2 or 3 bands close to the bottom of the gel. I don't think you looked at your actual DNA. If you didn't put it under any UV light, I think it's unlikely that you saw DNA. All right, let's take a break. You've earned it. And then we'll learn more. You really wanna? Well, for the Usabo, you need to know everything.
We have to learn here.
No.
I didn't study.
And I have,
You wanna learn it today?
there are like some houses that drop, they're dropping it.
I liked it.
Drop
I
exclusive
was like,
times. Tell them at the top of that, since you know it, you can. No, like you just gotta keep watering it. It's coming.
You'll learn to.
So forever.
Wadding is one with RNA, right?
What? We learn about bacteria. We'll learn about protists in chapter like 20.
There are a ton of questions about body systems.
The body systems? Physiology? Well, we haven't done physiology yet.
This one is like super easy once you get things.
Yeah, it's not difficult. It's very interesting. Okay.
Always wants to know that no sense of humor, you know?
No. So you know how when you cook a fish, depending on how you cook it, you can get like that, it's called aspic? No, what's it called? Like when, that's really gelatinous when it cools off. Listen, humans have for years noticed that when you do certain things to meats and bones, they get jiggly gelatinous stuff. And then you can use it for glue, or you can use it for very interesting foods.
A lot of gels are vegetarian.
All gels use them.
There are some gels that come from like seaweed and stuff.
Yeah. They're like egg roasts, right? Like there's no reason you couldn't eat that stuff. I think. Not when you add lithium bromide to it, you can't. Don't do it. Pretty much.
We have two, have not signed yet.
Okay, has anyone else signed in yet today?
Yeah, we have two people not signing yet. No one?
Are the numbers not matching up?
Yeah, we have 28 here, plus signing is only 26.
Oh, you don't sign in, right? She's TAing.
Oh, okay.
But, so that means one person hasn't signed in.
Yeah, okay.
You know, I don't sign in, right? Annika, you were requesting access for my 2018 Usabo. But you're in the 2019 Usabo.
Yeah, there's a 2018.
But then it's, I mean, it's very.
Bro.
Yeah.
But then we click on it and there's
And
like
it's
no
not,
access.
yeah.
You shouldn't have access to 2018
Then why do
videos.
Yeah.
Because I don't want to delete it from the folder.
But then like what if you want to watch it ahead?
Like what
You don't get the 2018 videos unless you pay for 2018 class.
You, if you want to study ahead of time, you can scroll and see what chapters.
That's true too.
Yeah, you can read ahead of time in the book.
Can you delete it?
No, I'm not gonna delete it. Because if I delete it, where's it gonna go?
You can probably make a copy of it.
Yeah, just make a copy. Why is it in our folder?
Dunno.
So you have the same folder everywhere.
Yeah, I don't want to create a new one. I just take some of the old stuff and put it away. The stuff that I need to keep from different year to year. And the stuff that's the same, I keep the same. The videos are not under my control, they're under the office's control, and that's why I don't want to delete stuff that's theirs, because I don't know if they have a backup anywhere. and I can't be bothered to sit down. I don't know, the organization's key, I'm not gonna mess with that. No, I remove access every year. Well, I mean, if you still want it next year, talk to me and I'll give you access to it. Yeah. But usually nobody's. Well, you know, usually like in 2020, you're thinking about other things. Like, it's very rare for somebody to be like, hey, give it back. Yeah, you can take advanced Usabo. And some people take advanced more than once. Yeah, because we cover a lot, and sometimes we don't do all the homework. I think there's a lot in it. It helps, but it's not.
It's like supplementary stuff. You need to know actually.
Yeah, you need to know the core. Like if you know the textbook, and then just keep thinking and practicing and reading, it's usually enough.
Oh yeah.
I think of advances more as an opportunity to learn more stuff.
I see.
It's a fancy way to say, we learn botany and physiology.
Just want to say hi. I don't even know if you remember me. You're lucky. You're lucky.
It is lucky.
Long story.
Microcentrifuge tubes too. It's just the little really little test tubes. They got little lids in you. I'll show you a picture.
Stuff in the lab. We have like a lot of
Yeah, they look like this. See? It's microcentrifuge tube.
Oh,
They actually look like that.
but there's a horizontal
Well, it's like that. It's like a little round, but it's not horizontal. You actually want the bottom to be as pointy as possible. This is your summer. I'm so excited. You're gonna come back and you're gonna be so nerdy.
Oh, you're like one of the younger.
You're sophomore, right? Yeah.
I used to go to Chinese class.
Yeah. In the Q building.
I don't remember his name.
He was a Chinese school. He's in my Spanish class last year. They always regret like 40 years.
Yeah, I celebrated.
Would you celebrate? You're celebrating the day that you were born. No, you're celebrating having survived another year.
You're celebrating becoming a year older. Anyways, for me, and go places. And also hang out with your friends when you're supposed to be studying.
You have New Year's?
Because they one high school student. And I know it's birthday party.
Well, they're making they're building a new study. Wait, do you know what they're coming up? The neighborhood that you see I used to live there and they have a new
He's slowly carrying all the
Yeah.
You know the what's it called? They must fire canyon at campfires.
Remember, it kills your brain cells.
And ran over a car.
In moderation.
Actually, the one has this one.
I don't like vomiting, and when you drink a lot, you vomit. Like, just don't do it at that point. It's alcohol poisoning.
or something like that. And then,
She just smells everything all the
yeah,
time.
but then she was saying that she's getting too full.
No, because it balances out the dehydration in the head.
She had fixed that.
These are she did. So she's all good. You find that there's someone like we send her.
That's why you see in things like Grey's Anatomy, where they're just like, oh,
I
come
don't know if you can.
on. TV show about doctors that's been around since I was in college.
Check out how there's
The
one.
other one's easier, but like not
Not
So you have no friends that has to stop.
See, that's pretty much what I wasn't allowed to as a teenager either. So that's pretty much all I did is watch a lot of TV.
Like she just takes out a bunch of it's like 5%.
let me give you
Basically, when you like lunch I don't know, it's like
We sleep is for those who don't have that. Okay guys, I have to teach you stuff again, even though we're all having a lot of fun talking. So, this electrophoresis, any questions? None? Good? Okay. So, in your textbook, it would be on the page opposite from PCR. So, there you are, it's on those pages. Please make sure you read that part for homework. Now let's rush ahead with various evolutionary stuffs.
Yay.
Yay! So in chapter 23, there was one thing two things I think that we haven't really talked about very much, which is modes of evolution and Hardy-Weinberg equilibrium.
So what? Party what?
Hardy-Weinberg.
Hardy-Weinberg.
You did read chapter 23, right?
No, I think, yeah. I mean, I did.
But it was a few days ago when you were sleeping, so you don't remember.
Well, my excuse is you said it too fast. So.
Okay, Hardy-Weinberg. You know what? At this point, I do not mind Hardy-Weinberg. But let's talk about modes of evolution real quick. So the idea is, they did a thing with the mice, remember? And they were talking about, well, do we have stabilizing? Do we have disruptive? Or do we have directional evolution happening? Or selection happening? Is this ringing a bell from the reading?
Yes.
The idea is, if this is the number of individuals with a trait, and this is the trait, then if we're talking about height, you know, most of us are more or less close to an average height, and very few of us are unusually tall or unusually short, right? Now, what if we were living in tunnels, and people who were too tall were more likely to bump their heads and die?
directional.
Then over time, we would have selection that would result in shorter people being more common, because the frequency of tall people would go down, the frequency of short people would go up, right? This is directional selection because it looks like it's moving in a certain direction on the histogram. However, let's say we're talking about, I don't know, giraffe and their long neck, and it's been a bad year, and it's been a drought, and so only the giraffes with very long necks have been able to reach leaves that other giraffes haven't. So what's going to happen this year is only the ones with long necks survive. The ones with short necks die and don't have children. So next year, we will have something that looks like this, right? The greatest frequency of giraffes will be the ones who survive the drought by having longer necks. Make sense? This is also, by the way, how Lamarckianism is different from Darwinism. Lamarck would say they were reaching for the leaves and they wanted to get them, and that's why they had longer necks. Darwin says the ones with these traits die, and so the ones with these traits became more frequent and were more likely to reproduce. What if we're talking about bunnies, and people really like the black bunnies because they're super goth, and people really like the white bunnies because, you know, magicians, but nobody likes the brown bunnies because they're kind of boring. So if this is white bunny, black bunny, brown bunny. And normally brown bunnies are the most common. What are you gonna have? Disruptive selection. And this would be an occasion of artificial selection causing disruptive selection, right? The humans are breeding the bunnies for sale at pet stores, right? So it's artificial selection. Finally, let me try to think of a sexual selection one. Okay, so if deer antlers are too small, the deer lose, oh yeah, sorry?
Can't disruptive change lead to speciation?
It could, but it's not necessarily likely to unless there is, unless the ones that are on two sides don't breed with each other, right? If like the white bunnies are still willing to breed with the black bunnies, then it's not going to cause speciation. All right, so sexual selection. You have deer, they have antlers. The deer with the small antlers lose all the fights and therefore cannot impress female deer and have offspring. The deer with too large antlers are just too heavy to pick up their heads, and so they also lose all the fights. So over time, you have only a small number or a limited size of antlers that tend to survive and reproduce, and that's called stabilizing selection. All right, let's talk Hardy-Weinberg. We ready? The idea behind Hardy-Weinberg is Punnett squares are thinking if this is one parent, maybe the parent had type A blood, and then this is the other parent, and the other parent had type AB blood. Then these are the four possible genotypes of the offspring of these two parents, because we're basically taking eggs versus sperm, right? Hardy-Weinberg says, what if you're looking not just at the genes of these two parents, the alleles of these two parents, but the genes or alleles of all the parents in the population? Assume random mating, that all individuals could potentially reproduce with all individuals. How can you predict their offspring? Let's say you only have two alleles. Hardy-Weinberg works for any number of alleles, but let's say you start with only two alleles. So let's say the frequency of the first allele plus the frequency of the second allele equals all of the alleles, right? Because there's only two. So that the frequency equals 1. P, frequency of one allele. Q, frequency of second allele. Since there's only two alleles together, we have frequency of 1. Then, the number of eggs total with this allele should be P, number of eggs. And the number of eggs, a number of sperm, again, should be P, number of sperm. Does that make sense? If the frequency of the allele in the population is 25%, then 25% of eggs and 25% of sperm should have it. All right, so yeah, let's put that down. So let's say 25%. So then what's going to be Q? 75%, right? 75. This is going to be 75. Okay, so what should be the frequency of individuals who inherited Q from dad and Q from mom? 75% times 75%. 9/16. Which is what percent is 9/16 about?
0.6.
It's like 50-ish.
56.25-ish percent? Okay, how about these individuals? A quarter times a quarter is an eighth, which is approximately what percent?
16.
About 12.5%.
16.
It's 1/16.
1/16, my bad. Which is
6.25%.
Okay, thank you. And then how about this person? This group of individuals? 75% times 25%.
316.
3/16, which is
18.
right? All we gotta do really is square the equation and we get p squared plus 2pq plus q squared equals 1. We're saying that if all the individuals in the population are having kids with themselves, within the population, then the frequency of homozygous dominant is p squared. The frequency of homozygous recessive is q squared. And the frequency of the heterozygotes is 2 times p times q. Does that make sense to everybody? And then the Hardy-Weinberg problems you're going to get are Hardy-Weinberg problems that maybe tell you how many individuals have this phenotype. So then you go backwards and you figure out how many would have that phenotype, you figure out what Q is, you figure out what P is, and then you can report their frequency in a population, right? At this point, if you know these two equations, and you know dominant versus recessive, there's a lot of math problems I can give you. Yes? What? Just in this population, I just named a number. Like maybe in this population of humans, the number of humans with the gene for 6 toes is 0.8%. It's less than 1%. It's 0.8%. So if you plug that in, and I tell you the gene for 6 toes is dominant, okay, so the gene for 6 toes is P. It's dominant, and it's 0.8%. Can you calculate to me how many humans in the human population will have 6 toes? Based on that? Should be p squared plus 2pq, right? So you can plug and chug. Yeah? Good. What other questions might you get? Here's the thing. Hardy-Weinberg is being taught to you in the evolution chapter for this reason. Hardy-Weinberg only applies if the frequencies of alleles don't change. Evolution, by definition, means frequencies of alleles are changing over time. That's the definition. Hardy-Weinberg, this math only works if there is no evolution. No sexual selection, no natural selection, no gene flow, no immigration, no emigration, no new mutations. Yes?
So is there a way we can like derive or make a new equation based on this equation for our alleles changing?
Yeah, that's the idea. The idea is this equation can give you the baseline if there was no evolution. And then you can actually count and figure out what's actually happening and compare the two and be like, based on this comparison, there is selection for a certain allele or selection against a certain allele. Yeah? Exactly. That's what we're learning this, to give us a baseline. And to give AP Bio tests questions they like to ask us about math. Yes?
So 2 questions. So one, I heard that the new API again is there's no like grid line question or something, like math grid line question, so it's like all embedded.
Since when?
This year?
This year?
They changed the format.
What?
If they changed the format of API this year.
What?
So we have
It changes the whole class.
What? I'm gonna have to change my class now? Seriously? Is there a new AP Bio format? No, it's my job to teach you to the format. Okay, well, I'm gonna Google this a little later and figure out if what you're saying is true. And probably and possibly change it.
Like 3 response questions or something.
It's just the number of free response questions?
I don't know. I think there's. I heard that it's changed to multiple questions.
Well, guys, it's exciting. It's a new day. It's just like what they did to us in 2013, where we've been teaching you a memorization-based exam for like 30 years, and then suddenly they were like, hey, it's not memorization based anymore, it's critical thinking. Psych! And we were like, well, teachers who cared redid their whole thing, and teachers who didn't care are just still giving you the same exams they did and are not preparing you for AP anymore. Okay, anyway, you're good with Hardy-Weinberg? Any questions on Hardy-Weinberg? I don't care if they don't have math questions anymore, you need to know Hardy-Weinberg. Usabo has it. Okay.
Does it say anything like college publications or like those here on college or just yeah, like they just change, I think they just changed the number of series ones to multiple choice graders and graded questions. And that's all of that.
Oh, nice. So there'll be more multiple choice math and less gridded. Okay, that's not a big change. Which means that you should publish a new practice test for us, which would be nice. Let's talk about chapter 24 real quick. Who read it? Nice, well done. So we should be able to go through it quickly because everybody kind of knows what's in it, right? What is the biological definition of a species? Somebody I haven't called on today, please. All right, yeah.
They can't produce fertile offspring with another group of organisms?
Yeah, so if the two organisms are the same species, they should be able to produce fertile offspring. If they're not, they shouldn't be able to. Exactly. So if, I find a bunch of purple sideways walking ants, and I think that they're related to some, brown sideways looking ants. I actually think they're the same species. That this is just a weird phenotype. How would I be able to prove it? Yes? Try to get them to mate with each other. And my next best option, look at their DNA. Why can't we just define different species based on DNA? Yeah? Yeah, there's a lot of genetic variation between individuals of the same species. For example, if you compare me and Henry, presumably Henry, you have a Y chromosome and I got 2 X's, so Henry is a whole different chromosome than me, right? That's a really big difference in genetics. Compare a male bee to a female bee. The male is haploid and the female is diploid. That's a humongous 200% difference in DNA, right? All right. So, are we all comfortable with what's happening on page 490? The reproductive barriers? Like, we're comfortable that if species literally live in places where they can't meet each other to reproduce, then they're not the same species. If one lives in the water and one lives on land, or if one is diurnal and one is nocturnal, or if one, so that would be what type of barrier? All right, temporal, that's right. So if one is awake in the day and one's awake at night, that's a temporal barrier. What if I'm fertile in spring and some other individual is fertile in fall? No, it's, yeah? Okay, so it's still temporal, it's different times of year. Don't let my tricky questions trick you. Question?
Yeah, so basically what you're saying is if I have two organisms, one of them lives in the water, one lives on land, but they can reproduce and have fertile offspring, they're still not the same species?
Well, do they reproduce and have fertile offspring in nature? Not if one lives in the water and one lives on land, they'll never meet.
Right, okay.
Yeah? Sure, if you take them out of the normal situation and put them in the same cage in the lab, they might still be able to reproduce.
Okay.
But naturally, if they don't reproduce, then they would be different species.
Oh.
Good question. So again, if one bird has one mating dance and another bird has a different mating dance, what kind of barrier is that? Reproductive. Mechanical isolation, fascinating subject, literally is trying to say that the reproductive organs don't fit together. Is that gonna apply if you have external reproduction? If your sperm and eggs meet out there in the water? No. So organisms with external reproduction are gonna be depending on, gametic isolation. Gametic isolation means the gametes have proteins that can lead to fertilization, and those proteins don't fit well with each other. And that is how sea urchins avoid replicating with or reproducing with the wrong sea urchins. Is there a question? No. Okay, just corner of the eye kind of thing. So even if you have reproduction, it doesn't mean the offspring survives. It could be non-viable or less viable. It could be infertile, like a mule, which is the offspring of a horse and a donkey. And you could have hybrid breakdown, which is over time, your hybrids, your grandkids and great-grandkids become smaller, weaker, and less viable. All right. In this chapter, I like to point out that ligers exist, lion-tiger hybrids. I do not believe they are fertile. Yeah, they have issues. I also want to point out that growler bears exist, grizzly and polar hybrids, and are getting more frequent with global warming because the places they live are now starting to overlap. So they used to be two different species, but they might fuse into the same species. Yes.
Are they fertile?
I think they are. I don't know. Google it. Yeah.
But over time, then would they become, if they aren't turned out right now, over time would they become fertile?
No, let's have a conversation about that. So in your textbooks, please flip to page 499, figure 42.14. Looks like this. 24.14, yes. On page 499. Stop coughing. Not good. All right. In this figure, we're talking about a species that has been separated from itself. Maybe like the grizzlies and the polar bears. They've been living in different environments and have over time gathered up many adaptations that make them ideal for those different environments. Now something has happened that means they're again in an overlap. Now sometimes you get that overlap naturally. We call it the hybrid zone, where two species overlap a little bit in where they live, and in that situation they have a little bit of hybridization happening. In that hybrid zone, you're going to have three possible resolutions. First, we have reinforcement. What do I mean? Let's say when a polar and a grizzly have a polar grizzly, right? A growler. If that growler is not very viable, then both the polar and the grizzly have invested a lot of time, effort, nutrients, hard work on an offspring that's actually not viable. That is going to create selection against hybridization. Basically, any polar who makes eyes at a grizzly is less likely to successfully reproduce. Any grizzly who makes eyes at a polar is less likely to reproduce successfully. And so the tendency to hybridize is just not going to be sustained in the gene pool. This is how all those isolations and barriers actually develop, right? They start having maybe, here, let's say this is the breeding season, okay, months, January to December. This is the number of bears that are reproducing. Let's say polar bears normally reproduce in these months. And grizzly bears normally reproduce in these months. But polar grizzlies are non-viable. So the ones that are reproducing during these months are going to die. They're not going to be able to pass on as much. The ones that reproduce during very different seasons are going to be more successful. And we're going to have directional selection for time of year of reproduction. That could be one way in which we introduce temporal isolation in these two species. Or maybe we'll end up introducing behavioral isolation. Maybe the grizzlies that are only picking on grizzly behaviors to reproduce with are more likely to have healthy offspring, right? And so that gets more frequent in the population. Does it make sense how, if we have non-viable offspring, we're going to have selection against hybridization and introduce new isolation mechanisms? Yeah? Awesome. So that is what we call reinforcement. Now let's look at the other option. Growler bear? Good. Growler bear happy. If your growler bear is happy, then you're actually going to be encouraging fusion of the two species. No reason for them to stop hybridizing. They'll keep hybridizing. The growler bears will keep hybridizing. You know, they'll just fuse together into one big happy species. Final option is the polar grizzly, right? The growler bear. It's just very infrequent. I wrote him tiny to indicate that it just doesn't happen very often. And if it doesn't happen very often, but the population of polars and the population of grizzlies is huge, then they're not really going to be affected by such a small number of hybridizations, even over great lengths of time. And so we will have maintenance of the status quo, which we call stability. Questions? All right. Yes. Stability. When, you know, the hybrids happen so infrequently that they don't really make a change to the species, to the two species. Now, who can tell me the difference between allopatric and sympatric speciation? I'm hoping somebody in the back, 2 rows. I will be mean and pick on you, yeah?
Isn't allopatric like geographically isolated?
Yes, allopatric means different country. Like Patrick, patriot. Allopatric means different country. Sympatric, same country. So allopatric's speciation means speciation because you're physically isolated from each other and you're developing new adaptations that over time mean you can't reproduce with each other anymore. Sympatric is weird, right? Sympatric is even though you're in the same location and could theoretically still reproduce with each other, you don't. What stuff makes sympatric speciation happen? Why would sympatric speciation even happen? Yeah? Sexual selection, right? If one group really, really likes, I don't know, tall individuals, and the tall individuals only like reproducing with tall individuals, and then the small individuals like reproducing with small individuals, over time, you're just going to end up with just 2 separate species that are not reproducing with each other, right? Another thing that sometimes happens is if there's 2 different sources of food, and like some individuals specialize for that one source and some individuals specialize for the other source, and them crossbreeding is actually counterproductive. The hybrids are not as good at either source and are going to die out. Then hybrids keep dying while the ones that breed within the group are surviving better over time. That can lead to that. Does that happen? Yeah. We have that great fruit fly experiment on page 495 where one smart woman breeds fruit flies for 40 generations, which, given that fruit flies take about 3 weeks to breed, is about 2 to 3 years of her life. I'm sure she is doing other experiments at the same time though. No, you don't have to be with your flies 24/7. They take care of themselves. Yeah, you just put them in the jar with some food, and then, you add more food later. Anyway, so they're breeding, they're happy, the fruit flies are doing their thing. And then after 40 generations, these only eat starch. These only get, what, sucrose? Something else? Maltose. She goes, okay, I'll take some females, I'll take some males. Will you breed with each other? You can't make us. We don't want to breed with each other. Oh, okay. Is it because you were eating different food, or is it because you were in different jars? Thank goodness I controlled my experiment. I had multiple maltose jars. So if I take from the 2 different maltose jars, oh, they're so willing to breed with each other? It wasn't that they were in 2 different jars, it's because they had, let's try it again with the starch. 2 different starch jars. Are they willing to breed with each other? Sure, totally, yeah. It's the starch-maltose combo where they don't want to breed with each other. That's what that data and that figure is showing you. Take a minute to confirm I'm right. Please take a minute to look at the experiment, and then we'll move on to something else. Again, it's page 495, figure 24.10, the fruit fly experiment. Questions about the wacky fruit fly experiment of awesome. All right. Big idea here is that A, she does some good control in the experiment. B, even though it's talking about how they are allopatric, that is, they're in different jars, you could pull that data and hypothesize that this could also happen sympatrically. I remember reading an article about 2 or 3 years ago showing that sympatric speciation happens a lot more often than we think, and statistically showing that. Plants sympatrically speciate even more often than animals, and part of the reason is because they can do allopolyploidy. Mrs. Webster, what's allopolyploidy? Allopolyploidy is shown on page 496. Guys, take a minute, figure out what's happening in allopolyploidy and see if you're willing to explain it to me. It's weird. So, who feels brave enough to explain what happens during allopolyploidy? Yeah, but I know you know it. Yeah, all right.
So you get like an uneven sort of number of chromosomes in like the new species, because one of them goes through like meiosis and its gamete has like one set. So because here they said like 2n is equal to 6 for like the red one, and they said n equals 3. So basically it has like one pair of one set of chromatids. But then you have another organism that doesn't go through meiosis properly. So instead of the chromosomes splitting, they stay the same. So when it hybridizes with the other, with the species or with the cell that didn't go through meiosis properly, you get like a new kind of species. With 5 or 10.
Yeah, you somehow end up with all the chromosomes from one species and all the chromosomes from another species in a brand new species that won't be able to mate with either of its parents, but can mate with itself, because plants can do that. This happens a lot weirdly enough, often some of the fruits and vegetables you eat are allopolyploids. Now you know. And doesn't it seem like really unlikely? But it's a big world and it's been many, many years, and sometimes unlikely things end up happening anyway. Like eukaryotes. Somehow we happen. All right. Last part of chapter 24 that I like to point out is the difference in the difference between punctuated equilibrium versus gradualism. So the idea is we, at this point, probably are all thinking of evolution as happening gradually. A mutation crops up, and if it's beneficial over time, it becomes more frequent in the population. And then maybe another mutation crops up, and then if it's beneficial, it becomes more frequent in the population. And so over time, we're gradually, as species, changing, gradually becoming different. However, when we look at the fossil record, we see one species that doesn't seem to change, and then another species that doesn't seem to change. For millions of years, nothing, and then over like 5,000 years, boom, a lot of changes, and it's like new species. So that's the idea of punctuated equilibrium. You have equilibrium, no change, punctuated by periods of fast change. Okay. So, who's right? Both are probably right for different species at different times in history. It is quite likely that if your environment is very stable, new alleles and so on just don't become frequent in your population because the environment is stable. There's no reason to change. In fact, being different is less effective than being the same as you were. Whereas when the environment is changing, like climate change, which happens over time on Earth, just is happening much quicker now because of human behavior, then what you end up having is a period where it's change or die. And so only organisms that are evolving quickly are likely to survive that. The other thing to consider is fossils don't show all the changes and differences over time, right? Fossils are like things that can survive millions of years in rocks. Bones, for example. Not things like hair color, right? We can't tell sometimes, okay, imagine two birds. Like in the front page of the chapter, we have those two birds that look identical, and yet they're two different species of bird. Imagine if those two became a fossil. Would you be able to tell that they're two different species of birds from looking at the fossil? No, the bone structure might be different, but I mean, look at how identical they look. The bone structure is probably pretty similar, right? You need some pretty big differences in bone structure to be like, oh, these are probably two different species, right? And so it's really hard from a fossil to tell how it's changing over time, whether it's speciating or not. Last thing I like to talk about is how long it takes for a new species to happen. What does the book say?
It depends.
Great answer. Ranges between about 4,000 and 40 million years, averaging at about 1 million. Yep. It's a big range. Let's take a slightly shorter break this time. Let's start again at... That watch hasn't been updated, right?
Yeah.
Cool. Okay. Good.
Oh, wait.
Yeah, we still have another hour of learning before us. No, I'm not that interesting.
I do try.
Yeah. I love Jane Fonda so much right now.
Who's Jane
They're trying to teach her not to demonstrate, huh?
Yeah,
She's
I guess in general.
actress.
she's like 80, she's like 86 podcasts a week. She's been podcasting outside music and investing everything. Why? She has my recently about against climate change.
Oh.
And because she has age and money and celebrity on her side, so she is using her privilege to make a difference, which is what you gotta do.
Oh, I
Yeah.
thought she was like, doing better than this for what?
No, no. The Frank protests are yeah. I'm very fond of him. You know, her first movie was Barbarella in the 60s. You will not enjoy it. I really genuinely think you have to be high to enjoy that movie, like people were in the 60s. A lot of the hippies were. And that was the intended audience of that movie.
Are you gonna change the clock?
No. I'm sorry. I'm not climbing up to change the clock. Good for her. Really proud of her.
She's so happy.
Oh, and she's in this Netflix show right now, Grace and Kelly. Grace and Kelly? No, Grace and something else.
Grace and something else.
But anyway, she's really good in that. Not Will and Grace, so that was a good show too. I think she's in Grace and Kelly. Really good show. Two women whose husbands left them for each other.
Wait, what?
So the husbands were partners at a law firm, and they decided to actually go in love with each other, not with their wives. And then the wives end up moving together as friends, and it's about all sorts of political stuff.
Oh, I thought it was like,
No, it wasn't. Nah, it was nah. Yeah, it's a really good show. But also, you probably have to wait till you're older to watch it. I don't know. It's like PG-13y.
PG-13 is
I think it's PG-13. It's not R. And they're all in their like 60s, 70s, and 80s, so mostly deals with heart attacks.
that's really bad.
At least in season 1.
Wait, did they get like younger or something?
No, they don't get younger, but they have more relationships. So there's more relationship-y stuff. You watch Outward.
I'm not allowed to. I saw the trailer and I really wanted.
You really shouldn't watch Outward yet. You're just too young. It's definitely R-rated. It's X-rated.
That's a thing.
Yeah, X is like really, really explicit. Which is, okay, my opinion. Why do we let you watch people die on TV, but not naked on TV? Like, it's okay to watch all the characters die horribly and bleed to death and there's torture, and that's barely even R-rated.
Yeah.
Oh, but, ooh, their butts? Oh, no, X-rated. Can't show people butts. Butts are bad. Butts are worse than death. Which I think is silly, personally. What?
The death is all CGI.
Yeah, but the butts are sometimes fake too. There's butt implants in there.
Okay, anyways.
Oh, it gets bad. Like, you know, it's such a soap opera type thing. But, you know, you watch it once, possibly twice, and then you're like done with it. It's not like Buffy or Friends. Buffy and Friends you can watch forever. Well, your season.
I watched some of it and I was just like this.
Yeah, I mean, it was fitting into what we really wanted to see in the 90s. But, like, try season 3. Just try it. And if you still don't like it, or try, well, yeah, no, I fast forward through those bits. Like the fighty bits, they're boring. All they do is just punch each other. I know who's gonna win. Buffy's gonna win. It's cool. It's done. Fast forwarding.
What's Buffy?
Wait, what else is there to watch?
Buffy the Vampire Slayer was a really good TV show.
What else is there to watch in a cartoon?
I mostly watch it for the quips.
They have like 2
Quips. Like when they're like saying jokes and being funny and just being clever.
I watched a random selfie like he's taken it.
Yeah, but I'm not proud of it.
The thing is, spin-off, that's better.
It goes so downhill. Oh, the originals is the same badness, just different same badness.
Better.
I've watched it. Well,
I watched the trailer and I was like,
I enjoy it more reading like the summaries or the transcripts than watching it.
The last one was really good, and then the last season, the original stuff really bad because like good writer left.
That's your problem right there.
It's also kind of sad.
Okay, 30 seconds, then I gotta start teaching again. What's a good show you kids are actually allowed to watch? You should watch Firefly. Firefly and the related it's Cowboys in Space, it's good.
yeah.
Good choices.
But it's not really crazy. No, it was like really crazy feminist. Like it's like trying to be.
Which one?
That one.
That one?
It's like trying to be really pushy.
I still want to watch it.
Yeah.
Is there such a thing as pushy?
No, but like it's just like everyone. So like in the trailer, it's just like everyone keeps on mistaking me for him, and then that's like supposed to be a really big problem. But the thing is, she's wearing his costume, and it's like, of course they're mistaking you for him.
That woman
Where is
in
it?
the show is actually cute.
Yeah.
She's played Jungle
Oh.
which is like super cool. And there's a lot of shows that
It's a superhero TV show. It feels like a superhero TV show. It does the superhero stuff. But yeah, it's got stuff on it that's pretty awesome. Here's my take on it. When you're trying to fight against the way society sees stuff, every small thing you do seems like a really big. Like, you know the data that shows that if there is more than 15% of women in the room, men think it's the majority of women in the room.
oh, seriously?
So if there's like 30% women in the room, men thinks women are speaking more than are more than 50%. If women speak up to 30% of the time, men think the women are participating more than the men are in the conversation. And it's because we have this expectation of one thing to be in society, and when you have something else, it seems like a lot. So
I get it, but like
I don't think it's like that argument you make right there, sure. But the bigger thing that there's multiple women with multiple personal agencies that are independent of any male characters, that is the thing that makes it a good show.
A lot of superhero TV shows like superhero stuff.
We don't have enough though, we need more.
Yeah.
I really enjoyed Wonder Woman.
Yeah, it was so good. They're doing this.
anything like they're really good. Either if it's like really good, like crazy good, or it just like goes down like there's nothing that like compares, or like it's slightly better, it's like
Alt life. Okay, shall we learn stuff again? So we are officially on chapter 25. And okay, here's the thing about chapter 25. It goes into the history of life as we know it, which is fascinating. And at the same time, how much of this are you going to get asked about on the AP? Only the first section. So I'm going to go into great detail about the first section. Chapter 25. So the first section of chapter 25 is about the origin of life. And then the rest of the chapter is very interesting. I'll talk about it briefly. But in terms of what you have to know for the AP, the next thing you have to know in chapter 25 is figure 25.9, page 517, which is endosymbiosis. You probably need to know mass extinctions, which are discussed on page 521. Oh, and you should really take a look at adaptive radiation on page 525. Oh, and all of section 526, 25.6, all of section 25.6. So section 25.1, section 25.6, and then those few figures I mentioned in the middle. So, origin of life. We weren't there, we don't know. However, we make some really great guesses, and our guesses include observing stuff right now to see if it could have happened back then. So we start with the Urey-Miller experiment, where we have some liquid and some old earth gases. We have some electricity and a little bit of heat to evaporate that water. Run that for 2 weeks and you end up with monomers, which are cool. Monomers are cool. Are monomers alive? No. And so to be alive, you need monomers to polymerize and then form cells, right? So the next experiment we got going on is splashing water with monomers onto hot clay. And when you splash that monomer water onto hot clay, you get spontaneous polymerization. Okay. So now we're imagining on old Earth, there's the puddle with like the sun and possibly the lightning, and you got some monomers, and then, you know, maybe it's splashing onto the nearby mud and it's becoming polymers. Now these polymers are floating around in the water, and if some of them are phospholipids, what do phospholipids naturally do in water? They form a lipid bilayer. Nice. So for forming that lipid bilayer, we're basically forming what is called a protocell, which is, it's like a vacuole, or like, it's like a membrane that's like not a cell yet because it doesn't really have a metabolism, but it could. It's like this pre-cell. And these protocells can sometimes bud off and create, like, you know, baby cells, like bubbles, can create 2 bubbles. And so, very coolly, well, at this point we've gotten to the bit that's a bit hardest to predict. Okay, so you got some protocells. You didn't have loud cars back then, though. So you got some protocells, you got some macromolecules. Now what? Well, it's not until you have inheritance that you can have life. Life has inheritable genetic information. And you've already learned just how complicated transcription and translation are. But it is possible to have, you guys remember how RNA can have a 3D structure because it can make hydrogen bonds with itself? So we have this hypothesis, this RNA world hypothesis. So the RNA world is the hypothesis that RNA sequences could have kind of created these self-replicating RNA molecules. We know that sometimes RNA can act like an enzyme. RNA that acts like an enzyme is called a ribozyme. And is that with 2 Y's or 1 Y? Ribozyme.
I think
I spelled it right. Okay, woo, ribozyme. R-I-B-O-Z-Y-M-E. Right, it's an RNA enzyme. RNA that can fold into something that catalyzes a reaction. And we're thinking, if this RNA sequence is making copies of itself, it's probably really bad at it. So probably a lot of the copies it makes of itself are no good. And some of the copies are as good, and some of the copies might be even better at self-replication. So the copies that are better at self-replication are being selected for. And the copies that are worse at self-replication are not. That's when you start heritable information, and that is when you start evolution. It's not live yet, but it can self-replicate. And maybe if it's stuck inside a protocell, right? If the RNA is inside a protocell, then other fragments would be nearer to it, and it will have an easier time replicating itself. Because fragments will be near it, it'll have help. And somehow, at that point, we think through selection and luck and time, we evolved the first single-celled organisms. You can see doubt in your eyes. I have doubt in my eyes as well. I don't think this is the perfect explanation. Scientists are debating this all the time. We have lots of research trying to figure it out. But bottom line, we weren't there. We just know somehow it happened. We can only suspect that it could have happened in this way. All right. So, in terms of history, it's helpful to know that Earth is about 4.5 billion years old. And that about, I think it was 4 billion years ago, let me double check. I think it was 4 billion years ago that we have the first evidence of single-celled organisms. Yeah, sorry, it's about 3.75 billion years ago. How can you find a fossil of a single-celled organism? Well, when prokaryotes grow like in layers on each other and in the mud can harden into little bubble shapes of predictable shape and structure of bubble that we associate with the presence of prokaryotes in the fossil records, right? There is, I believe, a picture of it in your textbook on page 511, near the bottom of the page. It's called a stromatolite, and basically it's like layers and layers of dead ancient prokaryotes that are our first evidence of the presence of single-celled organisms on Earth. Yes?
So it's like the black color, right?
Yeah, blackish brownish. Let's see, is there another picture anywhere in here? Yeah, no. It's wait. Right here at the bottom, this one. Okay? So now, the next major cellular innovation is photosynthesis. Because remember, up until this point, what are they eating? Pre-existing, pre-synthesized, spontaneously synthesized molecules, organic molecules. That's their source for organic substances. They're eating each other. It's not until you have photosynthesis that you have the de novo synthesis, the new synthesis of organic molecules by a living organism from nothing, from inorganic molecules. So photosynthesis is actually taking carbon and making it organic and making it available for all the other organisms to eat, which is cool, has a side effect of making oxygen. Oxygen is poisonous. A lot of organisms die horribly. Those that survive are either ones that hide away from the oxygen in anaerobic environments, or ones that can handle the oxidative stress. Oxygen is reactive. We have to have all sorts of enzymes to kind of protect us from it and unreact us in the presence of oxygen. Yes?
And isn't it like the reason we age because it produces oxidants in our body and stuff like that? Like it's kind of like the reason we're dying?
Part of the reason. Aging is a very interesting science. Yeah.
Wasn't oxygen the cause of the first mass extinction or so?
Yes. It's considered, yes. Now, eventually we have eukaryotes about 2-ish billion years ago. The first multicellular eukaryotes are only 1.5 billion years ago. We move on to land about 500 million years ago. Or a billion. A billion has a lot of zeros. How many zeros in a billion?
9.
9 zeros. When do humans come onto land? When do humans exist? 400,000. So your textbook says 200,000, but since your textbook was published, there has been some new discoveries. So the latest discovery I've heard of has us at 40,000. Sorry, 400,000. 400,000. So we have only been around, guys, for 400,000. That's barely even a blink. Exactly. We're not even a divot, okay? We're not even a crack in the history of time. Ants have been around for longer. Bees have been around for longer. Trees have been along for longer. Apes have been along for longer. Crocodiles have been around forever. We just kind of, and we can just as easily, is all I'm saying. Yes?
Does that 400,000 years ago refer to specifically Homo sapiens sapien or?
Homo sapiens. Not Neanderthals, not any of the others. There was a bunch of different hominid organisms, but humans, I think, is 400,000. Okay, at least based on an article I read recently. We have already discussed endosymbiosis. Remember when a prokaryote gets eaten by a eukaryote, instead of getting digested, becomes the mitochondria and the powerhouse of the cell? Okay, so I'm not going to go over it again. You guys all heard of Pangea? The idea is the continents of Earth are always shifting. It's why we have earthquakes. Yay, California. And when the continents push into each other or rub against each other, right? And so over time, sometimes the continents were all one big continent, Pangea. Why are there only marsupials in Australia? When Pangea split up, that's the only continent where the marsupials were not outcompeted by eutherians. Mass extinctions. If you're looking at the rate of extinction of species, it's more or less steady over time. And it increases in times of great change, because in times of great change, more species can't adapt quickly enough, right? When we have spikes in extinctions, we're talking about, when this giant meteor hit Earth and changed the weather and climate of Earth for a while as a result. We're talking about times in history where due to the movement of the plates, we had a lot of extra volcanoes, which changed the climate of Earth. We've had multiple ice ages, we've had multiple warm ages. We've had changes over time that sometimes result in a lot of death of a lot of species. But right now, scientists think, not all of them, but many think, that we are going through a 6th mass extinction. They have all sorts of evidence for this, but one piece of evidence I want to point out is figure 25.17. So, Albus, can you tell me, what is the x-axis of that figure showing you?
Are you looking at 25.5? Which one are you?
25.17, it says fossil extinctions and temperature is the title of the figure. Have you found it?
X is time.
Axis is the x-axis?
The X, yeah. 17.
Oh,
I was at 25.18.
You got it?
Yeah.
Okay, cool. What's the x-axis showing you?
Temperature.
Temperature. Is it literally the temperature of 0, -1, -2?
No.
No, it's difference from average, right? Good. And how about the y-axis?
Relative extinction rate of marine animals.
Relative extinction rate. And it is in the, okay, it's relative, so it's normalized. So when the extinction rate goes up twice the normal extinction rate, does that usually happen when Earth is warmer or when Earth is cooler? Warmer. And if Earth is warmer by like 2 or 3 degrees from normal, that is even more likely to happen as a mass extinction. Guys, we're already way past 1 degree average warmer than average due to global warming. We're past 1.5. We're heading there. We're on track.
Beautiful.
Any action that we take right now is trying to prevent us hitting 3. Right? We're really trying hard to prevent hitting 3. The Paris Agreement is about trying to prevent 2.5. And the point is, we don't want any of it, right? We don't want to even hit 2. We want to just prevent this, right? The less we change the average Earth temperature, the less we will be impacted. And all the species around us. The best way to prevent hitting that is national and international action to prevent the release of carbon and sequester more carbon. Now, every time you breathe, you release carbon. I'm not saying stop breathing. What I am saying is even how we farm, even how we take care of land, makes a difference in how much carbon is hidden versus how much carbon is available. When we use fossil fuels, it takes carbon that was underground for millions of years and puts it in the air. So what if we stop using fossil fuels? That'd be nice. That would reduce a lot of that carbon being released. Are there other options? Yes.
Nuclear.
We got solar energy, we got wind energy. Are we saying just use one or the other? No, there's lots of options. There is pyrolysis, which is cool. It's using trees, using a type of combustion of the trees, but then that carbon gets re-sequestered by trees. So it's not taking carbon from underground, taking only a certain amount of carbon, very carefully combusting it to reduce the total that's being released. A lot of that carbon is then available to still be used as fertilizer. It resequesters the carbon more effectively. So yeah, pyrolysis makes me interested. But we're totally off topic. There's a lot of ways we can do this. The number one way not to do this is to tell companies, yeah, do whatever you want. Because we have proven time and time again that humans are structured to prioritize short-term goals over long-term goals. When you're not given time to choose, you choose the short-term success over the long-term possibility of success, right? Do you want $100 right now or $102 next year? $100 right now. You don't want to wait next year for just $2 extra. That's not enough.
I mean, inflation is this.
Okay, 105. Beats inflation. Honestly, we just want the $100 right now. I want it right now. I could buy a really nice dinner for it. But yeah, however, we've also proven that when we sit down in a room and think together and talk and plan long term, we can figure out the right choices. And when it's a law, you don't have the choice to just make the quick best decision right now. You have to make the best long-term decision if it's a law. So all I'm saying is this is the kind of stuff we have to handle on a big scale.
And the Trump administration rolled back.
Let's not even think about the Trump administration right now or I will have a heart attack right here. They have rolled back everything, stuff we've spent years trying to put together to protect us. You know, air quality? Okay, if this is the amount of pollution in the air.
You got it going, okay.
You got me going. 2 seconds. Amount of air pollution in the air. Here's 2016. Trump gets elected. It goes down.
I want to watch this. I want to watch this.
And it's going up. He's just literally undone what we've been working for decades to do. He is literally making you less healthy right now by breathing. Okay. Now I'm done. Section 25.6. Read it. I don't think I need to go over it, but do read it. 25.6, please. It's got the bit about eyeballs. It's cool. Oh, and adaptive radiation. Okay, so here's what's going to happen once humans are gone. Or, at any point where you have a mass die-off, a mass extinction, or even when you have a seed that falls on a barren island in the middle of the ocean. Anytime an organism enters an environment of zero competition, that organism can radiate evolutionarily to fill lots of different roles. So, in the olden days, when dinosaurs ruled the earth, I'm bad at drawing dinosaurs. I thought I could draw dinosaurs.
Was that 3 minutes?
Okay. Here we go. It's a dinosaur. I drew it. Nails. Okay. It's a better dinosaur than I could have drawn 2 seconds ago. So anyway, dinosaurs ruled the earth, and then, oh no, it's a giant meteor. It squishes them, they're dead. No, it's not how they die. It changes the climate on Earth. It literally raises the temperature on Earth to like 400 degrees Celsius. Everything that's above ground dies. Eggs in mud, burrowing animals, they live. Well, guess what? Back when the dinosaurs were all over, mammals were little rat-like creatures. That's all mammals were. That's all mammals could be. If a mammal evolved to fill any other kind of role, well, there was already a dinosaur in that role. There was already pre-existing competition. However, with dinosaurs gone, mammals adaptively radiated to fill a lot of niches. We have flying mammals, we have swimming mammals, right? We have mammals that are predators and mammals that are prey, herbivores, carnivores, omnivores, right? They fill every niche because suddenly there was all these niches available. Do you guys know what I mean when I say niche?
Niche.
Yeah, niche. So, some people say niche,
Niche.
but I think that's weird. But you say what makes you happy. What I'm saying is habitat is where you live, niche is your role. Niche is how you live. Okay, it's only 2:30. Yeah, sorry, I'm flipping through your chapter for you. Yeah, we're good. We're done with chapter 25. We're going to talk about chapter 26 now. You get to read it this week. Oh my god. Why don't you read it? Well, this thing. Okay. We are going to discuss the science of naming things, the science of giving things names. Nomenclature. Well, actually, nomenclature is how we say the name of something in a fancy way. Taxonomy is how we group them. Systematics and phylogeny. Phylogeny is when we're grouping things based on how they evolved. So if I wanted to, I could systematically put all the flying things in one group and all the crawling things in another group. So Worms and snakes would be in the same group. Birds, bees, and bats would be in the same group. It could. Humans have in history. That would be one systematic way to organize the organisms and give them names. However, over the past 300-400 years, we have chosen to instead name organisms based on their relationship to other existing organisms, phylogeny, based on their evolutionary relationships. So, worms are more closely related to leeches and very, 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 very distantly related to snakes. Snakes are more closely related to reptiles, which are more closely related to mammals than they've ever been to worms. Make sense? Big picture? So, taxonomy is actually giving the things the names. We usually use Latin, and we use a very hierarchical structure. Our hierarchical structure basically groups all the organisms in a big group, and then those get grouped into smaller groups, and then those get grouped into smaller groups, and so on. The biggest group possible is a domain. Domain with an E? I don't know. And then you got kingdom, phylum, class, order, family, genus, and species. This is one of the easy memorized things to do. Just say it a few times and just go into your brain. Domain, kingdom, phylum, class, order, family, genus, species. Kingdom, phylum, class, order, family, genus, species. Did you say it a lot? Yeah, that's the other mnemonic you can use. So, dumb kids play chase on freeway, get squished.
That's very funny.
Or in my day, our teachers were not as dark humored, and so they told us that duh kings play chess on freshly grown sod. Duh kings play fresh. Listen, domain was new when I was a kid. It was usually just kings play chess on freshly grown sod, and then they came up with domain, and they're like, duh king. We were in Chicago. We spoke differently there. Dem kings? Dem kings play chess. Sod? It's like a type of like grass grows from the sod. Anywho, we got the hierarchy of taxonomy, right? Now, binomial nomenclature is just saying two-name naming. Binomial nomenclature just is one way of naming organisms based on the genus and the species, and that should give you enough information. Like, a lot of species have the same name. Like, there could be a species of frog and a species of bird that have the same species name. But, like, you combine genus and species, they're pretty unique enough. Always in Latin. Genus is always capitalized, species is always lowercase. And of course, because it's Latin, you italicize it. Anything in a foreign language is italicized. The rest of the chapter, we're going to talk about trees. Not the pretty kind of trees, though. Though these arguably have their own beauty. So we're going to talk about phylogenetic trees. Phylogenetic trees. So, have you just collected all the good markers?
They're just here.
A likely story.
Probably put them here.
Quite possibly. So, the idea with the phylogenetic tree is that a phylogenetic tree can act as a hypothesis about the phylogenetic relationships between organisms, meaning which species evolved from what species. The idea is where you have a node we think of that node as a common ancestor. Where you have branches, we think of them as the current species. Think of here, the endpoint, as being species that exist today. Any species branch that didn't make it all the way, extinct. Okay? It doesn't have to be drawn like this. It could be drawn like that. It could theoretically still be the same tree, depending on the number of nodes. This, I don't have to draw like that. I could draw it like this. I just spun something around a node. I could spin around this node, spin around that node, spin around this node. Like spinning around a node, it doesn't change the tree. Just because you're judging based on relationships to nodes. Does that make sense to everyone? Okay. Now, let's take a look, please, together at figure 26.4 on page 538. Figure 26.4 on page 538. It's like, got a tiger in it. I know. He's a kitty. All right. So, I just couldn't think of the word leopard. Okay. It was upside down. All right. I'm gonna ask you. Yeah. Ah. Homework back again. Well, you guys didn't pick it up last time. You left a lot of it behind. You know what? I'm gonna stop like 2 minutes before we're supposed to stop, so you guys have time to like pick it up. Okay? I think this is from this class, right? Okay. I don't know. I got 3 piles. They're all in the back. If your name is in one of these piles, pick it up. It's gonna like just hang out here next to Elvis, okay? Pick your homework up, please. Okay, now let's finish the chapter and then we'll be good to go. So looking at that figure, again, figure 26.4, we got some animals on it. True or false? Sorry, I need to sequence it up. The otter is as equally related to the badger as it is to the coyote. False. Obviously false, right? You can just count by the number of nodes. The otter and the badger are much more closely related. Okay. The leopard is equally related to the otter and the badger. True or false? True. Why is it true?
Same amount of nodes.
They're literally like the same distance from each other. So like, this is equally related to the ancestor species. Therefore, it's equally related to both of them. Okay? True or false? The leopard has been evolving for longer than the otter. False. They have been evolving for the same amount of time. They all started out as a prokaryote about 4 billion years ago and have been evolving ever since. All right. You are not more evolved than a bacteria. You just have different adaptations. The leopard is not more evolved than the otter, they just have different adaptations. Okay. The common ancestor of the otter and the badger existed earlier in time than the common ancestor of the dog and the wolf. Oh, sorry, of the coyote and the wolf. True. How can you tell? Because of the location, the location of the node. It's earlier in time. Right? How about the wolf is equally related to the panther and the otter?
What panther?
False. Which one is it more closely related to?
Oh, I think
The otter, the leopard, sorry. It's more closely related to the otter, right? Because they have a more recent common ancestor that they share. All right, here's what I want you to do. Grab either your phone or your calculator or your book bag or a piece of paper. Not your entire book bag, like your pencil case or whatever. And flipping to page 543, hide the tree from yourself using your phone, your extra notebook, a big eraser, whatever.
like the entire
Hide the tree from yourself. Page 543, figure 26.11.
The entire tree or just the end?
Now your job is to use the information to try and draw a tree that makes phylogenetic sense, that has a more closely related organism with a more recent ancestor. Try it. If you don't try, you'll never learn how to do it, right? Then you may leave the class. Yeah.
Is it 0.4 where we were looking at earlier?
Hide the tree.
Wait, hang on, which figure?
This one's figure 26.
Yep. Sorry if you have the wrong version of the book.
Where's the leopard?
Yeah? Yeah, that's that one. You got it.
So the tree but not the chart.
Cover the tree. Reveal the chart. Yes. Yeah, good. Cool. Sometimes it's easier, sometimes it's harder.
Did you eat chocolate?
I will show you the correct answer. But yeah, you can check it when you're done and see if you're right. Alright, what are my organisms? Leopard, turtle, frog, bass, lemprey. Leopard, frog, bass, something.
So it's kind of this, and I can see that I wasn't paying attention. See that too, What really related? Huh?
I know. But I'm good with that.
Oh, I was like literally
Unintentional. It's like
opposite.
It's like you read about 4-5.
Alright, here is why my answer is technically more correct than other answers.
I don't like it.
The leopard and the turtle have the most stuff in common, right? The leopard and the turtle have 4 characteristics in common. Therefore, their recent ancestor must have been recent in history. The leopard and the frog only have 3 things in common, so they must have had an ancestor earlier in history. The leopard and the bass even earlier in history, with the lamprey earlier, with the lancelet even earlier. Make sense? Notice that all the lines go to the end because all these organisms exist today. None of them have gone extinct. If I wanted to do it curvy, I could have done it curvy. Like that's okay. If I wanted to do it using a bunch of V's, I could have done it with a bunch of V's. Let me see if I can draw that here like this. And then that, that, and that. I could have drawn it like that if I wanted to. I guess if you really want to draw squigglies, it's up to you. But those are the three most common ways of drawing it. That all makes sense, right? AP will definitely give you a characteristics tree and ask you to draw a characteristics chart and ask you to draw a tree. The chart can show you stuff they have in common or stuff that they have that is different from each other. Look, for example, at figure, for me it's figure 26.14. It's the one with the human, the mushroom, and the tulip. Hide the tree, look at the table. Hide the tree, look at the table. Make sure you read the table legend, which says it's the percent difference. The table is showing you the percent difference. See if you can't draw a tree.
Just like I used to hit this.
Okay, some people have a version of a book without it. I'm sorry.
They do have it.
Where is it?
Flip more.
Flip more? More flipping, okay. Maybe. Possibly. I don't see it.
Wait, is that
Sorry.
I'm not responsible for that.
Here, I'll draw it on the board for people who don't have it.
It exists on the 11th.
Okay, for those who don't have it, we got mushroom, human, tulip. Mushroom, human, tulip. Now, mushroom is not different from mushroom, so there's zero difference. What's the difference between a mushroom and a human?
30%.
30%. And what's the difference between a mushroom and a tulip?
40.
40. What's the difference between a human and a human?
0.
0. How about a human and a mushroom? 30. A human and a tulip?
40.
40.
You
Yeah, I kind of switched how it's drawn, didn't I?
Yep.
It's okay. Same difference. Right? That would be 30. That would be 40. 40. And itself is 0. This is the worst possible way to have drawn this table, isn't it? It's better drawn in your textbook, but the important thing is that we're showing differences, and that's how we do differences. So then, how do you like to draw that table? What's more closely related? Yeah? Humans and mushrooms are most closely related because they have the least amount of differences. Means they have a more recent ancestor. Tulips will have to have a more distant ancestor. Done. That is probably the most correct way of drawing it. If you draw it another way, yeah, sure, it could theoretically be true, but it's a lot less likely to be true. We have these two ideas. The first one is parsimony. Parsimony, if you're parsimonious, you don't spend too much money. Parsimony in biology means you try to find the most simple solution. And the simple solution is likely the most correct solution. The other concept we get from this chapter is this idea of we want to group things in such a way that everything in the group is related and everything out of the group is less related. And we call that kind of grouping monoclinic. Let's double check. They show an example of it in figure 26.10. Can I just show you something?
Monocular.
Looks like this on
Molecular?
page 542.
Monocytotic?
And it's known as a monophyletic clade. So if everything in the group is related to everything in the group, we think of it as monophyletic, meaning it's in one phylogenetic group, one evolutionarily related group. So it's monophyletic group. We can also say it's a monophyletic clade. Yeah.
So in the diagram, like when they're sharing, like on the tree, when they're sharing, like for example, the human and the tulip, or the mushroom, how they like they said they split it up into 50%, 15%. Like what does that mean? It means they share like 15%, or they have 15% of their DNA is the same.
No, actually, like 70% is the same. 30% is different.
Okay.
And when they're splitting it, they're like, well, this one went 15% different in this direction, that one went 15% different in this direction. That's why it's 30.
Oh,
It's
okay.
bad. It's weird now. Don't worry about that. Thanks so much. So, let's say on my tree, I decided that these two, I was putting them in the same group. Maybe this is a snake and this is a worm, and I'm putting them in the same group. That is not considered monophyletic. Does that make sense why? Because I'm not including their common ancestor. Now, if I said animal, right? If I said animal, actually I'd be including all of these, right? Animal includes snakes and worms in the same group, monophyletically. We're including the ancestor and all the related offspring in that same group. But if I said creepy crawly things, that would be not monophyletic. That would only be the crawling things which have worms and snakes, but not the other things, like leeches or lizards. Does that make sense to everybody? Okay. So that's it for chapter 26. We have a few minutes left. What I like to do with the remaining time, let's just go ahead and do chapter 28 real quick. Chapter 28 doesn't have much to it. Yeah, let's just demolish chapter 28. So chapter 28 is the protists. In chapter 28, I recommend reading section 28.1, and I recommend looking at figure 28.3. Other than that, the rest of the chapter is fascinating and gooey and icky and frankly terrifying. But you really don't need to memorize it unless you really want to. At the end of the chapter, on page 598, there is a summary table, which I would also consider to be optional. But if you want to memorize something, you can always memorize that summary table. The thing I really want you to get from the entire chapter, that one thing I really want you to get the most, is on page 578, is figure 28.3, is this figure right here. This is what I most want you to get from this chapter. And I don't want you to memorize the entire figure, heaven forbid. That will just make your brains melt, right? Instead, what I want to talk about is how all eukaryotic organisms are related to each other. Right? You learn that as eukaryotes, we're dividing into animals, woohoo! Funguses, woohoo! Plants, woohoo! And protists. Except, protists is not a monophyletic group. If you look at that figure, you can see protists, some protists are related to plants. Some protists are related to funguses, and some protists are related to animals. And some protists are just really weird and different and not at all like plants or animals or funguses. They're just weird and different. And protists, therefore, include these guys, and this guy, and this guy, and these guys, and then all those guys. Whereas animals, funguses, and plants are clearly a monophyletic grouping. Each one of them is their own unique group. Doesn't take in stuff from any other group. Why do we have a monophyletic group in our phylogeny? Because of tradition, and we can't be bothered to change it anymore. It used to be, if it wasn't an animal, fungus, or plant, we would just stick it in the big miscellaneous box, protist. And so we still have all the protists in this big miscellaneous box, and then within that box, we have come up with some better names for them. Right? Like amoeboids, are those on there? Yeah. Uniconta, that's the amoeboids. Anyway, that's chapter 28. We're going to finish just a little bit early today because A, you got to pick up your homework, and B, you got a lot of reading to do. You are reading chapter 25, chapter 26, chapter 28, and again, start at chapter 20. We're going to finish the next 25.
Did you say to get started on chapter 20?
I can put it in the homework. So send it to you, I'll put it in the
No, Cupertino legislation about the environment stuff is really hard.
We ban we don't really care if we would call them, like, always call them in, and they still like love them.
Cupertino's legislation is a lot about that, and we had a petition if this is going to try to make road spread and then environmental sustainability commission is backing us, but basically the legislature is so I'll send you a link. Cool. That's when it's going to reach those things.
Yeah, that'll help. See ya. Okay, bye. Bye.
Can you also get to look at AP Bio soon? So it's okay.